So I'm a clinical geneticist. I see kiddos in clinic about 20% of the time. And I want to frame the discussion about the 1,000 Genomes Project and how we're using the data in the context of how can it help us in the clinical environment. And I want to do that by starting out by pointing out that the way we do clinical genetic testing is very different than the way we do other types of medical testing. This is my mom. She's 80. She had upper left quadrant pain. She went to her doctor. Her doctor may or may not have actually done a physical exam. He just took a really quick history, and then he ordered a bunch of labs and some imaging. What he did was he did everything he needed in order to get to an answer very quickly to solve this problem, or at least to get him to point him down the right path. I want you to contrast that with what I do in clinic. I might see a young child who has developmental delay. I'll take a 30-minute or longer history. I'll do a detailed exam that involves, rulers, measuring tape, family history, things like that. And then when I choose a genetic test, I get to choose one, right? I don't get to choose a test that gives me all the information I need. I have to choose a test that gives me maybe one piece of information. And sometimes that's a little stressful because there might be other mitigating factors, such as, say, the mom is pregnant with a male fetus at 12 weeks, and they want to know what are the chances that this child has the same condition that the other child has, right? So why are we in this situation? Why do we do this? And to give you the punchline early, we really shouldn't be doing this, right? We should be able to get every answer from a single test. So the way we do clinical genetic testing, at least in the US, and I think in many other places in the world, of course, is we do it in a stepwise fashion. You start with one simple test, maybe a microarray or maybe a repeat expansion testing for something like Fragile X. If that's negative, the family might come back and you might do a panel, although we're slowly trying to kill panels, and I think that's going well, actually. So then you might do something like exome or genome sequencing, and if that's negative, you really are kind of out of options, kind of like you're out of battery on the clicker, there it is. The only option might be to reanalyze the exome in a couple of years, reanalyze the genome. But why can't we do this in a single step? And the other challenge is at the end of this, the diagnostic rate is less than 50%. So half of the individuals that we see in clinic today that we suspect have a genetic condition, we can't tell them the underlying molecular reason for that condition. So as was, my lab focuses on, can we use a single test, long-read sequencing, to both reduce the time it takes to make a genetic diagnosis, to increase the rate of genetic diagnoses, and also reduce barriers to accessing genetic testing, because they are quite high in the United States and other places in the world as well. So it seems a little silly to talk about what is long-read sequencing here, but I was taught to give talks in a certain way, and one thing that you do is you always introduce your technology or what you do. And the way that I think about long-read sequencing is that it simplifies the analysis of complex genetic regions, right? If you imagine you have this repetitive region here, if you analyze this with short reads, you really don't know how many copies of the repeat are there. You don't know the structure. You don't know what's going on inside those repeats. Long reads simplify it by simply, in many cases, just spanning the length of the repetitive region itself. These are information-rich reads, so you don't just get, of course, the A's, G's, C's, and T's. You get the 5 MCs, the 5 HMCs, 6 MA, et cetera. This is important because we know that if we use other technologies like short-read sequencing, we miss a lot of things. One of those things that we miss is at least half of the structural variants that are present in our genome. So with long reads, you might detect anywhere from 20 to 30,000 structural variants. With short reads, you detect about half of that. So how is it that long reads could be used to simplify genetic testing? So this is the diagnostic pathway or guidelines for Beckwith-Wiedemann syndrome. We saw some examples of this condition with methylation just a moment ago. You basically start at the top. I don't know if the pointer works. It really doesn't. So you start by looking at DNA methylation at H19, IGF2, and then the antisense transcript in KCNQ1. With long reads, if you do whole genome, you can see the difference in methylation at all three of those positions, so you can get that first test out of the way. Without having the parents, you can phase this into the maternal and paternal haplotypes, and you can see that it follows the pattern that you expect, with the red reads, or the methylated reads, going to one haplotype, and the blue reads, or the unmethylated reads, going to the other haplotype. If this is either negative or if it's not informative, you can simply use the same data, you can back up, and you can look for things like copy number variants within this locus. So here I'm showing you a nearly 1 megabase region using reads that are not great, 15 KB or so. And we're actually able to phase all of these reads into the two different haplotypes. So we could look for things like inversions, deletions, other types of structural variants within this region. If that's not informative, or maybe it is informative, maybe you detected uniparental disomy. Someone got two copies from one parent, two copies of a chromosome from one parent. So here what I'm showing you is isodisomy. So on the graph, heterozygous SNPs are plotted on top, homozygous SNPs are on the bottom. If your entire chromosome arm is homozygous, then you got two copies of the same chromosome from one parent. You can then go on and you can look for things like polymorphisms in genes that are associated with the condition. So here's CDKN1C. It's a rare cause of Beckwith, but it does happen. So there's a polymorphism here that is pathogenic. So again, we used a single test, long-read sequencing, to replace essentially what it could be 4 or 5 different tests, right? So this is one sample collection, this is one trip to the lab for the family, this is one insurance pre-authorization, right? This changes the way that this works for families and in the clinic. So I want to give just a couple of examples of how we've used long-read sequencing to solve some particularly challenging clinical cases. And then I want to put that in the context or use that to show why the 1,000 genomes data is going to be so helpful. So if you think about reasons that the diagnostic rate might be as low as it is, you can probably divide it into 2 kind of general areas. We don't know the function of every gene, so we have incomplete gene phenotype relationships. And then there's variants that are difficult to detect or interpret. I'm not going to cover all of these just because of time. I'm just going to go through a couple of them. And the first example is that many genes are actually quite difficult to sequence. I learned this during residency. During genetics residency, I was on service and a kiddo came in with respiratory failure. She was put on ECMO. We were oxygenating her blood outside of her body. And we did what we typically do in this situation. We sent an exome. We could only send a duo in this case because the father wasn't available. They called us about a week later, and they said that she had a likely pathogenic variant in a gene called Haiden. I didn't know what Haiden was. Some of you might. It's a gene that essentially makes your cilia make their full rotation. So if you think about reasons that someone might have respiratory failure at birth, having a ciliopathy should be high on your differential. The only thing is that this is a recessive condition, and she only had a single variant, not 2. The interesting thing about this gene is that it's large, it's 460 or 500 or so kb, but it has like a 350 kb segmental duplication within it. The 2 base pair deletions she had was outside of that segmental duplication, so the question was how well were short reads evaluating the regions inside of the segmental duplication? The answer is not great. So this is data from NA12878. This is not from the kiddo, but you notice that over 2 of the exons, you have fairly poor coverage. So if she has a polymorphism there that's pathogenic, you're probably not going to see it. So what we wanted to know and what her family wanted to know was could we actually sequence it and could we confirm or exclude a ciliopathy? I think, more powerful part about this story that I typically don't add is that she did well. She went home. But her family remained really paranoid, very concerned about her. And when I was in the hospital one night working, I saw her name come up on the ED board, so I went down and talked to the family. And this is before we finished sequencing her. And they said, they were at home and she drank some milk and she coughed and choked and she turned a little blue, but kids do that, right? But they were so concerned that she had a ciliopathy that every time she did this, they brought her back to the hospital. She would get an x-ray, she would get evaluated, it would cost several $1,000s, it would take a lot of time. But they had that in their mind that she could have a ciliopathy, right? So we did long-read sequencing, we did targeted long-read sequencing, we did adaptive sampling. This is on an R9 flow cell, so the data doesn't look great. But it covered Haiden perfectly. And we were able to go through the gene and see that she didn't have a second variant in the gene. Just to confirm it, we sent cells to some collaborators in Germany, and they also confirmed that her cilia looked fine. I'll come back to it, but just notice that over the region where there is poor coverage with short reads, there is a SNP in that exon, but the question is what's the allele frequency of that SNP? The second set of reasons that the diagnostic rate might be so low is that there are many variants that we can't identify, specifically structural variants. I'm going to start with a fairly straightforward case. This is a case that was referred to us from an outside group. This is basically a kid who had prolonged bleeding. They sent a bleeding disorder panel, and it identified a single change in a gene called FGA, which is perfectly associated with the phenotype. But they didn't find a second variant. This is a recessive condition. So we recruited the kiddo to our study. We did adaptive sampling again, I think, in this case. And you can see that I have phased this into 2 different haplotypes. And here on haplotype 2, it's probably hard to see, but the green is the known maternally inherited stop. The question, of course, was what was the second variant? I hope everybody can see that the second variant is a 4 kb deletion that removes the first exon. It's pretty clear that this is pathogenic, right? You're not going to make the gene if you're missing the promoter in the first exon. Other types of variants are more challenging to classify or categorize, one of which is shown here. So segmental duplications we've talked about a little bit. These are regions of the genome that are larger than a kb, that are more than 90% identical to other regions of the genome. And sometimes they're tricky because they can be overlapping, right? So in a family that had Fanconi anemia, that had a couple of affected siblings, they had a long history of clinical testing that identified a single change in the gene FANCD2, but no second variant. So we did, again, adaptive sampling and found a 400 or so base pair deletion that removed an exon. That exon falls in that region where there are 2 segmental duplications. So you can imagine why it's so hard for short reads to evaluate this. But because it's hard for short reads to evaluate this, how common is this? This is actually an in-frame deletion of the exon. So would it actually qualify right away as being pathogenic? And then the final example I really like, we actually just got this case not too long ago. We just solved this case not too long ago. This is a kiddo, he's now 8, but he presented pretty early with a suspected glycogen storage disorder. He had a panel that identified a single change in a gene called AGL, but there was no second variant. He got a lot of testing. He got an array, it was negative. He got an exon level array, it was negative. He then had a research-based short-read whole genome sequencing by a small San Diego-based company. I won't say their name. They identified a structural variant in AGL, and they said it was a translocation. And the family was ecstatic. They had an answer, right? But it had to be clinically confirmed, so they did optical genome mapping, and it was negative. So what was the difference, or what was the disconnect? So we enrolled him in our long-read sequencing study. And what we found here, the known variant was on the maternal haplotype. The second variant is likely this ALU that's actually at the +2 position after the exon that we think disrupts splicing. But again, we have a problem. How do you prove, or what's the allele frequency of this ALU? What if it's 5 or 10%, right? It may not be detected in short-read sequencing. So of the 4 cases I showed you, there's 3 of them here that I really can't tell you what is the frequency of these variants. They could be more common. They could be common, they could be rare, but how do we use this to actually prioritize and categorize these variants? And that's where we turn to the 1,000 Genomes Project. So I think a lot of people know this. It's a collection of about 3,200 samples. It was started with HapMap, and it's been used extensively to evaluate patterns of normal human variation. One thing that's missing is long-read sequencing of this collection. So what we've done is we've started by selecting 800 of the 3,200 samples for long-read sequencing on the ONT platform. And our goal is to build a catalog of variants for filtering and prioritization. We want to really expand our understanding of what variation looks like in challenging regions of the genome. You know, what's the allele frequency of variants within PMS2, for example? And then that'll also allow us to evaluate variants in regions of the genome that are associated with really interesting signals, like with RNA sequencing or other types of data that exist for this collection. Our approach is, I would say, fairly simple. We take the cells, we grow them up, we extract the DNA. We run 2 different pipelines. We do an alignment and an assembly-based pipeline. The assembly-based pipeline is the CARD pipeline, so it's Shasta. We do variant calling from the alignment and from the assembly. If I had to bet, I think the variants are going to be more useful from the assembly-based pipeline, but I'm not sure what Fritz thinks. Probably the alignment-based pipeline, or do you have an opinion? No? Yeah, from both? And our goal is to generate data at least 30x average with average read lengths of at least 50 kb. This is data from the first 100 samples, and on the y-axis you can see the average read length, so we do get as high as 80 kb. And then each dot, or there's 100 dots on here along the x-axis. We froze the first 100 samples. These are all done on R9 flow cells, and all this data is available right now on AWS. So if you want to kill the hotel Wi-Fi, you can go to AWS right now and start downloading it. You can see that when we call variants, we see the patterns that we expect. So insertions are on your right, deletions are on your left. You can see the ALU peak on top, and you can see the line peak on the bottom for both insertions and deletions. And then you can see that the novel structural variants follow the pattern that we expect. Again, this is 100 samples. So you can see that as you add individuals, you get more and more novel structural variants. And then you get that bump when you hit the African samples, where there are more novel structural variants in those samples. So what can we do with the data? So this is an application that was built by Sophie Gibson. She's a graduate student in the lab. I will say that being a PI is terrible most of the time, but working with graduate students makes it worthwhile, especially when they surprise you one morning with an application that lets you browse the 1,000 genomes data just by clicking. So what she did is, it's a Shiny app. We're going to convert it to something a little bit more useful. But what you can do is you can take any repeat expansion in the genome. You can see what it looks like in the 1,000 genome sample. So you can click on any one of those the plots, and it'll tell you which sample has the number of repeats within that bin. And then soon you'll be able to actually see the reads in IGV from the website. So let's go back to the ALU insertion that we found. And the question is basically, is this rare or is it common? Gus Gustafson, he's another graduate student in the lab. He built a filtering application for genes. It's under development. If you'll notice, the end variance there is, I think, greater than the number of atoms in the universe, so that's not right. But this is very new, so we're working on it. But basically, you can look through for any gene and see what the allele frequency is for variation within that gene. And this is rare. I'll just close by pointing out that I really think long reads is going to change how we do clinical genetic testing, and it's going to improve outcomes for everybody. I think we're going to see, and I say this a lot, we're going to see a single test that will replace our current stepwise testing approach. We'll see it for both cancer and germline testing. We're going to have variation called from haplotype resolved assemblies. That's going to increase the diagnostic rate, shorten the time to diagnosis. And then I do think, and I didn't talk about it, clinical ultra-rapid sequencing is going to be quite standard at academic centers. The things we do are going to reduce barriers to accessing testing to where anyone in the world can have access to this, essentially comprehensive testing within a couple of hours. And I think it's going to make a big difference in low resource settings, regions where it's difficult to get testing in, things like that. So I'll stop there. I want to say thank you to my lab. They're really the reason that I get anything done these days. There's a lot of folks in the 1,000 Genomes Consortium. It is open to anyone. So if you are interested, just scan the QR code. We have a Slack group that you can join. And then there's several folks from ONT that I didn't acknowledge on here, but for example, Dan Fordham, Andrew Markham, who are quite active and helpful within the consortium. So with that, I will stop and say thank you and happy to take any questions.